I always do that.
People inside.
Okay.
Guys, we are going to start. Okay, let's kind of get started.
Are we good with the live stream?
Huh?
Are we good with the live stream?
Are we okay with the live stream? Okay, great.
Awesome.
Should we start? Yeah? Okay, good. Let's start. Yeah, it's a pleasure to have Cesar here, all the way from Chile.
Yeah.
Yeah, he's from Catholic University, right?
Yeah, the Catholic
Okay,
University.
so talk about science.
I'm going to talk
Research
about
and
things.
academia.
Yeah, I'm going to tell you a little bit about my story. So, hi, I'm Cesar, and I'm kind of a protein nerd, and I will tell you a little bit about how I got here, including coming to India. But before that, I just want to mention yesterday by Frederick and also by Leo today, which is that this is an EMBO Global Lecture Series. And so EMBO is a great program. India is also included as one of the associate member states, as well as Chile. There are 31 member states and 4 in cooperation agreements. India is one of them. Chile is also one of them. And so we can basically not only network with more than 2,000 members, but also we can find sources of funding for fellowships, for postdoctoral researchers, scientific exchange grants, different funding for courses, workshops, and lectures. And also there's financing for different projects that you might want to pursue as being part of the Joint Investigator Program or the Global Investigator Network, of which I am a member. And the other thing is that EMBO is not only like a community for networking and having grants, but also for publishing. So they have really cool journals that I wish that I can ever publish there sometime. And they also have like a peer review platform that is pretty good for actually achieving that goal of publishing. And so now I'm going to move on to my talk after that commercial break. And I'm going to tell you a little bit about research from the end of the world. So before we start about that, I'm a professor, associate professor from the Catholic University in Chile. I'm also an adjunct researcher from another, what they call Millennium Institute for Integrative Biology, which is like an institute of frontiers in Chile. And I'm also an EMBO Global Investigator Network member, okay? So Chile, right? Coming from very far. Chile does not come from Chile. That's just for starters. Chile comes from, people believe that it comes from a word from the natives, that it means the end of the world or the end of the land, okay? Not a Chile. And it has beautiful landscapes, but you might know us by other things, which is earthquakes, tsunamis, and volcanoes, which are typical in the news. They happen, but not so frequent, so if you want to visit the country, please do. The probabilities of you experiencing one of them is pretty low, but it's not zero. Okay, so that's a picture of me, a recent one. Like I said, I'm associate professor from the Catholic University of Chile. I'm also part of a Millennium Institute of Research. And recently, member of the Latin American Federation for Biophysical Societies, Executive Counselor of Protein Society, EMBO member, a member of the Rosetta Commons, which is a community of computational protein designers. So we design proteins using computers. I'm director of a graduate program, and I do too many things. And I don't know how I got here. And so for the reasons of this conference, in which I'm very grateful to be invited by India Bioscience, I have to tell you how I got here. So I have to do like a quick recap of how I got here, okay? So let's look first at the research. So if you're interested in the research that we're doing, Like I said, we work on proteins. We are protein nerds, so we work on 2 different projects. We work on proteins that can change their structure dramatically to convey different functions to proteins. So this is paradigmatic because we didn't imagine proteins would do that. And so we use that to try to understand how proteins actually command novel structures and novel functions. And we also study proteins that can degrade plastics, which are enzymes. So enzymes that can actually eat on plastic and whether we can use them for biotechnology. Okay? So this is me. I also work out. That's like a side project that I have. I go to the gym. I also do hiking. I love to do hiking. Something about 8 years ago, I was not a faculty member. I was a researcher at another university, the one that is like on the competition from the one that I'm right now. And I also did weightlifting and a bunch of hiking. Even before that, way younger and like 20 kilos less than now, I was working on enzymology, not on protein biophysics and protein design, and I was in a band. And even before that, when I was like an undergrad, I was thinner, and I was doing immunology and plant science. I was not doing proteins. And so at this point, you might have some questions. Did I meet any rock star? Yes, I did. I met Chris Cornell, Duran Duran. I met Nine Inch Nails. That's a picture with Romy Fink, which is a guitar player from Nine Inch Nails. Second question that you might have, where did the hair go? I have no idea. I'm getting bald, and it's extremely bad. And the other questions that you might have in terms of research is, how did you decide to switch to proteins? How did you figure out that you wanted to work on proteins that switch structures and that degrade plastics? And how did you work on like having now all of these so-called accolades of being like a member of too many things? And so I'm going to try to explain that by going to the very beginning, which was my transition from being an undergrad to a PhD. And I met this guy. This is one of my best friends in the whole world. Company in Germany. He learned cryo-EM, but by 2007, he was a lab mate, or he was becoming a lab mate of me. By 2007, we were a little bit crazier, so that's a picture. If he's watching this live stream, this is going to be awful. He doesn't know that I'm showing this. So Felipe is the guy that is opening the mouth. We were having too many drinks, I guess. And I used to work on plants. So I used to work on like root development of carrots under light and dark conditions. And Felipe and I met in a conference. And he showed me this, which is that he was doing phylogenetic analysis of enzymes that were doing different catalytic reactions, but they were from the same family. And he was using computers for that. And I was blown away, because I wanted to do the same. So after the conference that we both ended up by random chance, I joined the lab. And the first take-home message that I have is, don't be afraid to explore a different path of what you're imagining that you're going to do. Plant science was not for me. It was proteins. And so this random encounter actually had a career-changing event in my scientific life. So I started my PhD. I started working with these proteins that are this collection of proteins that we are showing on the screens. These are proteins that can form oligomers in different ways, in different shapes. And I wanted to understand how. And I also wanted to understand whether the proteins will come together and fold at the same time, or if folding happens first, or if folding happens afterwards. I wanted to understand that process, so learning a lot of biophysics. And so I met my supervisor, Victoria. She was a great supervisor because she was working on enzymes from the functional perspective, but she was really supportive, and this is important, supportive of my ideas. I wanted to do biophysics, let's do biophysics. But she didn't have the tools in the lab for doing it. And so we paired up with a professor in Sao Paulo, Richard Garrat, to actually do the experiments because he had the availability of all of this. Was a great combination. Something happened. And what happened was that I was studying at the University of Chile, and the University of Chile had an agreement with the University of California in San Diego. And the agreement was, we have this big pot of money for sending people to UC San Diego for at least 6 months. And I did not have any idea of what to do, but the money sounded good. And so I did it. I applied for it. I applied for that opportunity. And so I started looking for people that I can work with, and I found out the perfect supervisor, Betsy. or Elizabeth Comides, for the people that are not familiar with her. She works on protein-protein interactions, so one thing that is great. She oversaw the biophysics facility from UC San Diego, which was also great. And the third thing, she had a great technique that I wanted to learn about, which was hydrogen deuterium exchange mass spectrometry. For those that don't know about mass spectrometry, you can measure peptides or molecules of different kinds of different sizes. You can determine their size. And so the idea of this technique is that you can use deuterium, which is heavy water, as a probe of solvent accessibility in a protein, and you can chop your protein into pieces, and then you can study each piece, and you can determine this part is solvent accessible, this part is not, and you can understand better your protein structure. What I did not know by that time is that in front of the building that she was located, there was another building that was called Urey Hall, which harbored a center for theoretical biological physics, pure computational stuff. And it was led by these guys that are here, Peter Wallines and Jose Onuchik. And these guys created something that I read through many papers, but I was not aware of, which is the protein folding theory. And this was the key thing that I was trying to apply on my thesis. And so something that happened out of random was that by taking this opportunity, that I never thought it was about the professors, it was about the money, actually led me to collaborate with all of them. And this had an everlasting impact on my career. If we look at the articles that we have published so far, I have published 10 articles with Betsy to this date. I have published 7 articles using the techniques that Jose developed in his lab, including one article with him, my first co-author article of my life. And I also published 2 articles recently with Peter Wallines. So all of these collaborations actually meant something now. And many, many visits to UCSD. I still travel to UC San Diego to actually do experiments myself. So second take-home message for all of you, it's embrace all opportunities and collaborations. Even those that sometimes do not make that much sense, they can bring you some really cool opportunities if you take them. All right, so now I had to decide on my own adventure what I wanted to do as a researcher, right? And this was in my mind, like stuck forever, the phrase that I'm showing on the screen right now, because it was told by a friend of mine. And he was like, people do not care about protein folding. They just care about the final protein structure, because that's what explains function. So, I mean, that's it. And it's cellular function, so proteins don't matter that much. And I was like really devastated by that. But at the same time, I was like very intrigued whether I can find something that will actually mean something different for people. That now protein folding is important, and now protein function is also relevant outside the cellular context. And that's how I found the proteins that I showed you before. Fold switching proteins, or metamorphic proteins, for those who are familiar with the term, are proteins that can switch their structures to encode different functions. So what we're showing on the screen, which is a blue region and an orange region on the left side, are actually the same protein with the same sequence, just different structures for different functions. And the idea is that these processes are critical for function. If they do not happen, there's no function. So protein folding is unavoidable. You need it now for the cell to actually do what it's supposed to do. And they might explain how proteins emerge in nature. So it's like a crucial, fundamental science problem. And then on the right side, we have the plastic degrading enzymes, very famous. They degrade man-made polymers. We don't know whether they happen because we contaminated too much, or they just happen by chance. But we know that if we engineer them, there's opportunity for biotechnology. And so I found my 2 different models that were like, now I'm responding the question that I want to solve, protein folding and protein function, but I'm finding something that other researchers might find useful, not only myself. Okay? So I'm going to skip the examples because it doesn't matter. I'm just going to show you how cool they are in terms of plastic degrading enzymes. This is a video with a collaboration with Germany, in which we show that if we take one of the enzymes and we put it at high temperature and we add a plastic tray made of PET, the thing degrades in 24 hours. So that's what we're aiming for, right? Okay, so we did a bunch of work. We have published in many articles. You know the drill. But I was so excited about this, about trying to pursue this project, that I tried to get a grant without having a faculty position. I strongly do not recommend this. It's so bad for you. So I was stressed out. Like I was like working in some kind of like postdoctoral position at the University of Chile, and I had this grant now that I had to work on, but I couldn't get PhD students because I was not a faculty member. So I can get master students and undergrads, but no PhD students. And I had to work on this. And on top of that, I had to do the postdoctoral research work. So it was a struggle. And so I started applying for positions. And I was rejected in many of them, because this is not how things work. You get rejected. It's part of it. So I applied to different universities in Chile, rejected, or sometimes shortlisted, interviewed, and then rejected. And I also applied for positions elsewhere. I think in France I tried once, maybe in Portugal I tried another time. Didn't work out. And then something happened. Again, something happened out of just luck, which is that 3 universities called for positions for professors in either biochemistry or computational biology in the same year. In the same year. And I was there for that. So I applied for them, and I got offers from all 3 of them. And I am not saying this for bragging. I am saying this for saying, this is rare. This does not happen frequently. And I was lucky to be there. So the other lesson is that I moved my lab to the Catholic University of Chile because I thought they had better support, which they do. It was a new environment. I wanted to work on something different, like with different people, working on different research. But it was a lot of work. I was prepared for this. I felt prepared. But it was an equal amount of luck. If that did not happen, I wouldn't have gotten the position. Okay? So it's not, sometimes you might feel frustrated because you're trying hard to get a position, and then you get a no. There are multiple factors for that. It's not that you're not adequate for the position. Sometimes it's like they're searching for something else. They're searching for something very specific in the department. The competition is just too high. There are many other factors that can actually chip in you not having the success that you wanted to have. And so you require a lot of work, but you also require an amount of luck that is not irrelevant, and you need to be in the right place at the right time. Okay? All right, so the position was great, moved with my lab, but it was a computational one, which meant I didn't have lab space. And I work on experiments and computations, so I was like struggling, like what can I do? So I have friends, Mario and Loreto, that actually helped me out. They said, we want to also have our lab, so let's share a lab space. And so we did. The lab was empty, and it might happen the same in India, maybe not. You have the space, you don't have the instruments. So you have to buy the instruments, and for that you need grants. Fortunately, I had grants, because I was a moron, and I got the grants before being a faculty member. So I had grants. So we worked on setting up the lab. The lab was great, a little bit small, but great. And then they made this other building in 2017, which was like even more impactful. This is the STEM building. We are located on the 7th floor. We're not using the whole building. There's no way that we can do that. But you can see the pictures from like the opening day, all great. But can you tell something from the pictures? Like beyond the idea that the photo of the mountains is actually the Andes that you can see from the balcony, which is great. But can you see something noticeable in the photos? Empty. So this was a signal. You need to buy the instruments again. Which means, go for your grants. Go for your grants. So I had to get grants again. And like I was saying before, for faculty positions, also grants, sometimes, they just take time. So when I got to the university, I got these 2 research grants. They lasted until right after I incorporated, like a year after. And then I started applying for grants to get the money for actually funding my lab. And I did not get funded. And I tried for all of those. So this is very frustrating. So what would you do if you were in this position? This is important to think about. Like, what are your alternatives? What would you do afterwards? And so we had an option. We were doing experiments, but we were also doing computational stuff. So we can still do a lot of computation. That's not that expensive. So we can collaborate. Maybe what we have to do is that in challenging times, seek support from your peers. Ask for their help. Ask them what they're doing, and try to help them out. Or ask them for them helping you out with the research that you were pursuing. And so this is a shout out for all of the people that we actually collaborated during this time. We made a lot of collaborations with people from the United States and from Germany. So we have Betsy, we have Irina at Ohio State University, Marcos at Ohio State University, Wolfgang Zimmerman that works on plastic degrading enzymes, Lynn Camerlin, now at Georgia Tech, working on computational simulations of proteins. People from the region, so from Universidad de Buenos Aires, Diego Ferreiro and Ernesto Roman from Argentina, Richard, again, from Sao Paulo, Ezequiel and Eduardo from Chile. We collaborated with a bunch of people. And this was great for surviving the lack of funding, because then afterwards, when we had a lot of results, a lot of preliminary results to show up, and a lot of publications to show that we're a good lab for people to actually join us, we got funded. All of those grants. So whenever you're struggling, don't stop. Try to do something else. I'm going to hear the ring. Whenever you're struggling with support, just try to find your peers and try to collaborate with them and try to get things done. Then I also became an EMBO member, a Rosetta Commons member, and we got the Paper of the Year award from Biophysical Journal in 2020. We also got, in 2022, we got information that we were one of the most read articles from Biophysical Journal, and I recently got the award for excellence in teaching from my university. I know that I have 5 minutes. Great, thank you. We have published 40 articles since we actually changed institutions. Of them, 38 are with at least one collaborator. And we have been invited to give 13 different talks in different conferences. So this is all because we started collaborating. This is not because we were on our own. This is not my own merit or credit. This is a collaborative effort. Our lab grew bigger. We're now 13 people. PhD students are 9. The rest are undergrads, and we have 1 lab manager. And so the other and last take-home message that I have is that the path to success is not a straight line at all. It's always challenging. You have to make decisions sometimes that are very hard. You have to change paths. You have to change your thinking about your project. You have to be humble to realize that other people are not understanding what you're trying to communicate and that you have to make changes. And that's part of being a scientist. For other things, for completing this game of being a faculty member, you also need to consolidate your lab somehow. And that means that you also need to learn from your own experience and from others how to mentor your students. And so we have this retreat, this biennial lab retreat that we do at the Marine Coastal Center from the university, in which we try to align expectations. We try to say, this is what I want as a PI, this is what you want as a student, and so let's try to meet in the middle. Let's try to see what you want to achieve, let's try to see what I want to achieve. And we try to then compromise and adapt and try to do that. And so some stuff that is important from this slide is that, or at least for me is important, is that it's not only of importance of the researcher, the type of research that we're pursuing. You need to check whether the student actually wants to pursue the same. Don't get a student that doesn't want to do your research, that just it's in need. Like, try to encourage him or her to actually get something done that is actually entertaining for them or motivating. Align expectations is really important, and also provide the time and effort for the students also to bring their own ideas and exploring them. This is really important for them. So these are pictures from the Coastal Center on the left. In the middle and on the right, it's the 2 different retreats that we have had for discussing very openly what are the struggles and challenges from the lab and what are their strengths. And so by that, we consolidate what we think is a very good and healthy lab culture. And since we just already started doing great with grants and with meeting with friends and collaborators, then we started doing other things for other people. We did a lot of outreach. So we created something that we call the Protein Biophysics at the End of the Workshop/conference that took place in Chile twice, Uruguay and Argentina. It was attended by something about 100 people. It had, in the beginning, just invitees from the region, but then afterwards it had invitees from other countries, and it was just great. So I encourage you, if you have the time for actually putting the effort to consolidate a conference on a topic that is of your interest, do it. And more recently we did this, which is even crazier, because we were in the position of actually doing it, which is that we run the first Latin American conference on artificial intelligence for protein design, which is the type of research that we're doing now. And what is important about this is that it's supported by EMBO. And also, by industry, and this was great. We got money from industry because the industry is also very involved in trying to get into the biotechnology progress. And artificial intelligence for protein design is one of the kind of spheres to actually explore the use of artificial intelligence in biotechnology. And so we got funding from AI Proteins, a company which is located in Boston and with whom we collaborate now a little bit. Cura Biotech, which is a biotech company in Chile, a bunch of universities, different scientific societies, and also we got even sponsored. So the last logo on the right, it's a beer company. And we got beers for everybody during the conference, so it was great, right? All I'm saying is that the paths for science are not a straight line, again. And so the takeaways from this presentation, I've already mentioned. They are over here. Do not be afraid to change paths if you have to. It's always important to do something different if you want to learn it even. Try to embrace all opportunities that face you, even those ones that look like, do you need to think about it, but like still consider them. Landing a faculty position, it's hard, requires a lot of work, but also a lot of luck. You have to be there at the right time, in the right place for it to happen. In challenging times, go ask for help. You will need it. You don't want to be alone when you're frustrated. And again, it's not a straight line. And just to complete this, when I showed this slide, I was telling you about all of the things that I do aside of the work. And I do that because I think this is really important. This is not something that you just do. This is important for your health. And this is the last take-home message. Whatever you do, whatever it is, always be kind with yourself. If you're struggling, like myself, so I suffer from severe depression, which is not easy. And it's like a taboo thing to speak about, but I don't feel like it's a taboo, so I just speak about it. Mental health is like complicated, especially if you're frustrated. And so you need to take care of yourself. So resting and making time for you and for others to do things, it's not outside of the work. It's not a reward. It's part of it. You need to be well rested, and you need to do things for yourself before you actually can achieve something important. And so these are the pictures of all of the friends that I have across the world, which go with me in adventures of going, I don't know, at Universal Studios or hiking or visiting different countries. And that's why I can be like standing here. It's not only about the science, but I can be standing here because I have friends that actually help me out, venting out what I need to, okay? With that, this is like the end of my talk, but before we go, if you're interested in going to a Protein Society meeting, this is the time for doing it. So this is kind of a promotion, free for them because I'm a counselor member. There's a deadline for open talks for the annual symposium and for travel awards. So if you do something about proteins or even outside proteins, maybe DNA or RNA, I remember there were a few posters about it. You might think about applying for the travel awards before April 1st and go to that wonderful meeting in Vancouver, Canada. And the other thing is that if you know students that still don't have a PhD, send them over to my country. We have openings for PhD students. We have even a joint degree program with King's College in London for PhD students. And if you're a postdoc, we also have fellowships from the government. And we are now in the process of application for postdocs until April 15th. Thank you for the invitation again, and looking forward for your questions.
Thank you. Thank you, Andrew. Cesar, for a very good talk. Very nice talk. It's nice to see your journey from being a rock star to be a rocking scientist.
Yeah, yeah.
Really
I should have stayed as a rock star.
Fantastic. So yeah, we have time for a couple of questions, I think. Yeah. Yes, one here.
Great talk.
Thanks.
I wanted to ask about metamorphic proteins. So what is the definition, like what are these, why they are metamorphosizing, and how do you compare this with its dynamics?
Great.
So what exactly is it happening?
This is a great question because I can go back to the slides that I skipped. Okay, so metamorphic proteins. Metamorphic proteins are proteins that can dramatically change structures. And what I mean by that is that, here is an example, our favorite protein. It's a bacterial protein, it's a transcription factor, it's called RFAH. So on the picture, what we have is what happens when you have transcription in bacteria. You have a protein that is in green, that binds to RNA polymerase and activates transcription, but not so much. Just the enough for encoding genes that are like very proximal in the operon, but not the ones that are at the very end. Those happen to be terminated. But there's a metamorphic protein, which is on the bottom, which you can see that there's an N-terminal domain, which is called NTD on the image, that it looks similar to the one on the green protein, but then there's another region that is called C-terminal domain, or CTD, that is like an helix. It's not like a barrel, right? So that protein is inactive if you have it that way. It needs an transforms into the same green. So that's a metamorphic protein. It's activated to transition to a completely different structure in terms of secondary and tertiary structure, and that is a requirement for function. If you don't have that transition, then there's no function. And so what we have done by doing computational analysis and also experiments is that we have explored what are the potential folding pathways or refolding pathways of metamorphic proteins. So the video is showing simulation of the protein going from the state in red to the state in blue, which is from helices to a beta barrel afterwards. And so that's pretty much what a metamorphic protein is. Different metamorphic proteins have different changes, but overall the definition is the same. Dramatic change in structure required for function.
This side, sir.
Okay, thanks.
One more question.
Okay, one last quick one. Yeah, okay.
Hi.
I'm highly interested in your work, whatever work you're presenting, the protein folding and this one. Can you studies about the protein repairing systems was there not?
Protein what?
Protein repairing system.
Protein depending system.
Repairing.
Oh,
like
repairing.
sometimes the protein, that is like kind of
Yeah. So
new things.
we don't work with protein repairing systems, but what I can tell you is that people have checked whether metamorphic proteins switch to one state and then they get deleted, like degraded in order for synthesize and then work again. And some works have demonstrated that they actually don't need that. The proteins go to one state and then they go back. And then they go over and over again. And they can do this many, many times. But different proteins have different time scales. So there are ones that are very slow. There's one that regulates circadian cycle in cyanobacteria. Extremely slow. Takes hours for actually the transition to happen. But this one takes a few seconds. So different proteins have different behaviors. Yeah.
Okay, thank you. Yeah. Thanks, Cesar, again. Yeah, for the great talk.
Awesome. Thank you.
Perfect. Thank you. Yeah. Okay, so let's move on to our final session in this on the day before we go for the walk and the cultural program. So this is about Ask Us Anything, and this is basically a session without any curriculum, but we still have something can talk. And there is a panelist. We have panel of 8 mentors and people. We have Abhijit, Andrew, and Vijay, Anandita, and Ritu, Roop, and Sunando, right? Did I miss someone? Maybe I did.
And this is a
and Ramre, yeah.
That's right.
Yeah.
And
Okay, so this is a panel discussion, but the panels are all dispersed.
Yes.
So they're all sitting
And they will
wherever
all
they're
sit
sitting,
where
so
they
we don't
are.
really have a